So spectrophotometry is a method to measure how much a chemical substance absorbs light by measuring the intensity of light as a beam of light passes through the sample solution. So the basic principles in that is that every chemical compound absorbs, transmits, or reflects light, electromagnetic radiation, over a certain range of wavelength. And then this measurement can all be used to measure the amount of a known chemical substance. So a spectrophotometer is an instrument that measures the amount of photons, the intensity of light that gets absorbed after it passes through a sample solution. And then with the spectrophotometer, the amount of a known chemical substance, its concentration can also be determined by measuring the intensity of light detected. And then depending on the range of wavelength of light source, it is usually classified into two different types. We got ultraviolet, visible, and the infrared regions. So the UV visible spectrophotometer uses light over the ultraviolet range, which is the 185 to 400 nanometers, and the visible range, which is the 400 to like 700 nanometers on the electromagnetic spectrum. And then the IR spectrophotometer, the infrared, uses light over the infrared range, which is from 700 to 50,000 nanometers on the electromagnetic radiation spectrum. So in visible spectrophotometry, the greater the concentration of absorbed ions or molecules in solution, the greater the absorption of visible electromagnetic radiation. So the absorption or the transmission of a certain substance can be determined by the observed color. For instance, a solution sample that absorbs light over all ranges of the visible spectrum, i.e. it transmits none of the visible wavelengths, appears black in theory. And on the other hand, if all visible wavelengths are transmitted, i.e. the sample absorbs nothing, the solution sample would appear white. All right, so we said that the degree of absorbed radiation is measured using an experiment or an instrument called a spectrophotometer, which measures transmitted light intensities. So the wavelength where the absorbing ions or molecules has a maximum absorbance of visible radiation is determined first and then set on the spectrophotometer for the analysis. So visible spectrophotometers in practice, they use a prism to narrow down a certain range of wavelengths to filter out other wavelengths that this particular beam of light passes through a solution sample. And then this is just an image taken from experiment 34, not 35, because it kind of gets into the theory behind all this. All right, so this slide basically illustrates, in the cartoon illustration, the fundamental structure of spectrophotometers. So it consists of a light source, a collimator, basically a lens, a monochromator, basically a prism, a wavelength selector, which is just a slit, and then a cuvette for sample solutions. Those cuvettes kind of important. These characters are kind of important. We'll get into that later. And then a photoelectric, sometimes called a photosensitive detector. Here it's just called a detector. And then some sort of display meter, right? So basically one needs a desired wavelength of light to pass through the solution sample, which would be in the cuvette. And then the amount of photons that are absorbed get detected by the spectrophotometer. All right, so one needs the spectrophotometer to produce a variety of wavelengths because obviously different compounds absorb best at different wavelengths. For example, we have para-nitrophenol, the acidic form. Looking at this absorbance versus wavelength plot, has a maximum absorbance at approximately 320 nanometers. And here, para-nitrophenolate, the basic form, absorbs best at the 400 nanometers, as shown on the slide. So we have two different peaks for the maximum absorbance of these two different compounds. All right, so there are several factors that control the amount of electromagnetic radiation by energy that a sample absorbs. So one would be the concentration of the absorbing substance. So that would be C. And then the thickness of the sample containing the absorbing substance. And this is basically determined by the width of the cuvette. Here it's called the path length. And then the probability of light absorbing substance. So this is called the molar absorptivity coefficient. It's got a lot of names. It's called the molar absorptivity coefficient, also known as the extinction coefficient, also known here as the molar attenuation coefficient. But I think we're just going to stick with the molar absorptivity coefficient, because I think that's what your lab manual says. But we'll talk about that later. All right, so the ratio of the intensity of transmitted light. Got something in the chat, one second. Back. There we go. Was the cuvette width one of the variables? Yes, the cuvette width was the path length. We're going to get into that later. That was just like, we're just barely touching on that. Okay, so let's talk about transmittance. Oh, we got something else. Thank you. So the ratio of the intensity of the transmitted light, here shown as I subscript T, to that of the incident light, I subscript O or 0, is called the transmittance, which is capital T, of the electromagnetic radiation of that sample. So once you know the intensity of light after it passes through the cuvette, after a cuvette, you can relate to transmittance T. So with that said, let's just rehash this. Transmittance is the fraction of light that passes through the sample. And then we can also, we also said that this can be calculated using the equation on the slide, where I subscript T is the light intensity after the beam of light passes through the cuvette, and I subscript 0 or O is the light intensity before the beam of light passes through the cuvette, as you can see here. This is before it passes, and this is after it passes. And then so just to repeat myself, transmittance is the intensity of light I subscript T over I subscript 0. So the intensity of light after it passes through the cuvette over the intensity of light before it passes through the cuvette. And then most spectrophotometers have a transmittance scale. However, chemists, or we would perform calculations based on the amount of light that is absorbed by the sample, because absorbance is directly proportional to the concentration of the absorbing substance of this unknown sample. So let's talk about absorbance. Okay, so with transmittance, we can get absorbance because transmittance is related to absorbance by the first negative log expression on the slide, right here. So absorbance is equal to the negative log transmittance, as you can see right here, which is equal to the negative log intensity of light after passing through the cuvette over the intensity of light before passing through the cuvette. Okay. Or we can say absorbance is equal to just the log of the I subscript 0 over I subscript T. So they just inverse when you do the negative log, okay? So this is just with no negative in front of it. Either way, absorbance it basically equates to being molar absorptivity, shown here as A, times thickness of the cuvette, shown here as B. It was called path length in the previous slide. And then C, which is the concentration, which is also, so all this would be all known as Beer's law. Okay, so we said that the concentration of an unknown sample in the cuvette can be obtained by utilizing the absorbance value that is solved from the equation and then using the Beer-Lambert law. So let's get into that law. Okay, so Beer's law states that there is a linear relationship between the absorbance and the concentration of a sample. So for this reason, Beer's law can only be applied when there is a linear relationship, when there's only like a straight line on the graph. And then the molar extinction coefficient, traditionally symbolized by the epsilon character. It's kind of fuzzy here, sorry. I think in your lab manual it's symbolized by just the letter A, lowercase a. But so this molar extinction coefficient is given as a constant and it varies for each molecule. So it is a measure of how strongly a chemical species or substance absorbs light at a particular wavelength. An important thing about it is that it's an intrinsic property of a chemical species that is dependent on basically their unique chemical composition and structure. And then since absorbance does not get any units, the units of the molar extinction coefficient must cancel out the units of length and concentration, which is seen as the letters B and C in Beer's law equation. So as a result, the molar extinction coefficient has the units of liters times moles raised to -1 power times centimeters raised to -1 power. And then the path length is measured in centimeters always. So the path length would be B. And then that's just because a standard spectrophotometer uses a cuvette that is like 1 centimeter in width. So it's always going to be equal to 1 centimeter. And then since absorption and the path length are known, we can calculate the concentration of the sample, given the absorbance as well. All right, so for this quick example, we'll be getting the concentration of a sample of guanosine, which is a purine nucleoside, similar to guanine, which is one of the 4 main nucleotide bases found in DNA or RNA. So for this particular example, we are given its molar extinction coefficient, which is equal to 8400. And on the slide, it says molarity subscript -1, but remember, molarity is just equal to moles over liter, and the negative just inverses that, so it is equal to liters over mole. And remember, this molar extinction coefficient is an intrinsic value to each chemical species, so it's basically just a lookup value. And then we are given the absorbance at 0.7, which was presumably determined experimentally. And then the path length of the cuvette is always 1 centimeter. So now we just plug in those known values into Beer's law equation, isolate concentration, solve for C, and we get this value here, 8.33 times 10 to the -5 power moles of liters. All right, so we talked about how transmittance relates to absorbance and how absorbance relates to concentration by using Beer's law. And then Beer's law shows us the absorbance value is directly proportional to the molar concentration of the absorbing substance of the unknown sample, but only if the same cuvette and a set wavelength are used for all of the measurements. And then if one plots absorbance versus concentration, we should get a linear function, right? And then the slope and absorbance data resulting from that plot can be used to determine the molar concentration of the sample absorbent species in a solution of unknown concentration based on that linear relationship. One sec. Right, so I was talking about the linear relationship. So this means that you'll have one set of solutions with a known concentration being used to prepare a calibration curve, and then a plot, which is basically just a plot of the absorbance versus the concentration. And then a second set of solutions will be prepared, and you'll determine their molar concentration using their absorbance data, which should be emailed to you. And then the calibration curve you just made with the set of solutions that you know their molar concentration. Okay, so to get the concentration of an unknown, we have to remember that we should get the max absorbance first to provide the lowest detection limits for this unknown. All right, we'll talk about that later. And then the second step of the process is to generate a calibration curve. So the calibration curve is generated by preparing a series of solutions, usually 3 to 5, with known concentrations of the species being measured. Okay, that's important. The concentrations are known. And then every standard curve is generated using a blank. The blank is just some solution that is assumed to have an absorbance value of 0 or close to 0. And it is used to zero the spectrophotometer before measuring the absorbance of the standard and unknown solutions. So the absorbance of each standard sample at the maximum absorbance is measured and then plotted as a function of concentration. Okay, and then the plot of data should be linear, and then assuming a linear standard curve is obtained, the equation will provide the equation that provides the best linear fit to the data is generated. So you're just going to get a function for a straight line, y equals mx plus b. What the heck? One second. I have no idea why the chat is not working. All right. How do you find the volume of the unknown solution? That was a question I got in the chat. So we're going to get to that later, but basically the unknown solution is going to be diluted. So the volume is not the part you should worry about. The concentration is the part that's going to be the tricky part. Okay. So where was I? Okay. So we ended up getting a linear function, a straight line. And then you should note that the slope of the line of the calibration curve is equal to the molar extinction coefficient times b, the cuvette length, in the Beer's law equation. Let me repeat that. So the slope of this line that you will get after plotting your data points will be equal to the molar extinction coefficient times the path length or the cuvette length. And then third step is to measure the absorbance of the samples with an unknown concentration. And then the absorbance of samples the absorbance of the sample that is used with the equation from the line in the calibration curve to calculate the concentration of the unknown, and you do that by solving for x, as in y equals mx, to get the concentration. Okay, so let's go over the actual lab procedure for right now. So first, in part A, you would prepare a stock of solutions by transferring copper sulfate to a volumetric flask half filled with nitric acid, and you would add about 10 mils of concentrated ammonia, and then you would dilute it to the mark on the flask with 0.1 mole nitric acid. So basically, obviously not gonna do this, but there's a video on Canvas to watch demonstrating how to do a dilution like this using a volumetric flask. And then if one was in lab, you would note the color of the copper sulfate, its state, is it like liquid, solid, etc. And then note the amount of copper salt that was added, as well as the color of nitric acid, stuff like that. And then part B, you would just skip that part. So in the past, we just gave you guys 610 nanometers as the max absorbance if using a copper analyte. And then in this actual video, the second video that you should watch on Canvas, the TA found the maximum absorbance to be about 400 nanometers when she was using a nickel-based analyte. So part B, we're skipping that, and we're just going to be using these two values based on if it's nickel or copper. And then for part C, you would set the spectrophotometer to the max wavelength. Then the absorbance measurements would be taken at the beginning with the blank. And then you would get the absorbances for the rest of the samples. But first, you would subtract the blank's absorbance from the sample absorbance before actually applying the calibration curve. And then here we have a modified table. As you guys can see, it's different from the volumes of the metal ion compared to table 35.2 in your lab manual. Then the volumes have been decreased. So you're going to want to use different volumes than the one in your lab manual. And that's because the ones in your lab manual are too high, and at a too high concentration, the absorbances may not result in a linear function. Okay, so the standard solutions are going to be using, the first is a blank of 0, and then 1, 2, 5, 7, and 10 mils. See if this works this time. Do we change the volumes in our report sheet? Yes, you do. So in your report sheet, I think in your report sheet you have what, 0, 1, 5, 10, 15, and 20, right? Just change that to 0, 1, 2, 5, 7, and 10. And make sure to include that change into your calculations when you're thinking about getting your concentration. Okay. I really wish this chat worked when I was, oh, nice. So will we only fill out part A and C and all the post-lab questions? Yeah, you're only gonna be doing part A, C, and D, because we haven't gotten to D yet, as well as the post-lab questions. And then you should be making a graph for this calibration curve. Okay, so we got one more in the chat. In the data we received, it said we needed to explain why we chose a certain wavelength, as in why you chose the maximum wavelength. Just, I mean, I'm gonna be grading you guys as just report sheets, so I think so. It wasn't too clear. Just tell them the values. I mean, I don't think there's even a section as to. I'm telling you to skip part B entirely, so just ignore that. You don't have to worry about that. Yeah. And then using the volume. Here, where was I? So yeah, we changed the volumes, we made them smaller, and then using the volumes of the stock solution sample, as well as the molarity, along with the volume you dilated to, you can calculate the diluted molar concentration, the molar concentrations for these diluted samples. Okay, and I think like number two part C in your pre-lab assignment basically shows you how to do that. And then the absorbances that Lee emailed to you, right? You use those two sets of values, the absorbances, as well as the molar concentration for the diluted samples that you yourself have to calculate, which you did do an example in number 2.C in your pre-lab assignment. With those, you can make your calibration curve. let's see. Yeah, absorbance have been emailed to. Yeah. And then solve for molar concentration using the volumes and molarities in the procedure. Yes. And then plot the absorbances versus the molar concentration for the standard solutions made to get your calibration curve. Okay, cool. Yeah, so make sure you guys include that calibration curve plot on Excel or whatever you guys use. Upload that along with your report sheet and post lab questions. That also counts for your guys' grade. I need to see that graph. And then for part D, you would collect like 30 mils of an unknown solution, and you would filter it if it's cloudy, and then you would place 20 mils of that sample into a flask, and then you would add like 1 to 2 mils of a complexing agent, and then you would dilute it to 25 mils using a volumetric flask with 0.1 molar nitric acid. And then if you were in lab, you would observe and note the color of the unknown solution, like if it was cloudy or not, what color was it, et cetera, et cetera. Oh, let's back up. So that's basically it. With your calibration curve, you can basically just get the absorbances of that are given to you of these unknowns, and basically using your calibration curve, find what concentration it is. So let's see. Yeah, the slides are going to be posted as usual. I wonder why the last slide was this, because this is old. Oh, okay, yeah. Let me just wrap it up with this. So you guys do not have lab next week. I don't know why. And then you have one virtual lab simulation going over Beer's Law. And then what's going to be due next Tuesday, even though we're not going to be meeting, is you guys' experiment 35 report sheets, along with the post-lab questions and along with the graph for that calibration curve. Okay. And then experiment 36 pre-lab assignment, we can just make that due on February 23rd, to keep it more fresh in your guys' mind. All the post-lab questions, yes. Okay, so that's pretty much it. You're welcome. I'll stick around if you guys have any questions. You're welcome. Sorry the chat was buggy. Yeah, the only thing that's going to be due next Tuesday is the report sheet, the lab simulation, and the post-lab questions and that graph. The pre-lab assignment is not due until the 23rd.
Thank you.
Mm-hmm.
Thank you.
Thank you.
Thank you. Thank you.
I have a question. For the
Yes.
lab